So peptidoglycan synthesis starts within the cytoplasm with the association of the nucleotide uridine diphosphate to the two molecules NAG and NAM. These molecules are the building blocks of the peptidoglycan wall. In order for these molecules to cross the membrane layer, they need a transporter. In this case, our transporter is bactoprenol. Now, before bactoprenol can transport both NAG and NAM across the membrane, the two molecules need to be attached. In order to do this, both the UDP molecules donate their phosphate group to bactoprenol. Once these phosphate groups are attached, they form a pyrophosphate bond which binds both the NAM and the NAG together. Once they form the NAG-NAM pentapeptide, it is able to cross the membrane layer through bactoprenol. Once in the periplasmic space, it is up to the autolysins, which is a particular group of the penicillin binding proteins, to separate the existing bonds between the peptidoglycan wall. Once these bonds are separated, the new NAM-NAG complex is allowed to insert itself into the wall and form a new piece of cell wall.
So now we're going to transition into the effects of antibiotics on peptidoglycan synthesis. And the first one we're going to discuss today is penicillin, which was first discovered in 1928 by a Scottish scientist by the name of Alexander Fleming, and people began using it to treat infections in 1942. The structure of penicillin resembles the terminal D-allenyl D-alanine, which is found on the peptide side chain of the peptidoglycan subunit. As you can see, the structural similarities presented on the slide. The structural similarities blocks the enzyme which catalyzes the transpeptidization reaction that forms the peptidoglycan cross-links. And as a result, the formation of a complete cell wall is blocked, which leads to lysis due to osmotic pressure. And it's also worth noting that the crucial feature of penicillin, which blocks the formation of the peptidoglycan crosslinks, is the beta-lactam ring, which you can also see present on the slide.
So here we can see the structure of our next antibiotic, bacitracin. This antibiotic can be used to prevent infections and minor cuts and burns, usually applied topically and not through an intravenous injection. So bacitracin is functionally different from penicillin in that it does not target the enzyme responsible for the formation of the peptide bridge between peptidoglycan bonds, but it actually targets the carrier, bactoprenol. In order for bactoprenol to be reactivated in order to transport more NAM and NAG molecules across the membrane, it needs to lose one of its phosphate groups and go back to the other side of the cytoplasmic side of the membrane. What bacitracin does is it goes and binds to the bactoprenol and prevents it from losing its phosphate groups, which prevents it from crossing the membrane again.